Next up, we have Nima Ahmadkhani. And Nima is going to tell us about saving more lives, how cryopreservation could end organ shortages. Welcome, Nima, to the stage.
Met Tanya. She was 28 years old when this photo was taken. Talented poet and mental health advocate, full of life and creativity. Behind Tanya's lovely smile was a battle. Tanya had been diagnosed for a kidney failure and spent over 3 years waiting for kidney that never came. Sadly, Tanya became one of the 17 people who dies each day in the United States while waiting for an organ transplant. But what's more surprising is knowing the fact that the problem is not organ availability. In fact, the number of donated organs far exceeds what we need for transplantation. The problem is time. For example, for a heart and lung, we only have about 5 hours preservation time before transplantation. Therefore, 70% of donated hearts and 81% of donated lungs get discarded without any use. And this is not the end of the story. Even when transplants are successful, in half of the cases, they get rejected within 10 years of transplantation due to the chronic rejection, which again emphasizes a problem, which is time, because there is not enough time in order to making the recipient body ready to receive a new organ, which can prevent most of these chronic rejections. But the good news is my research, which is on cryopreservation, can solve this problem. Cryopreservation is a long-term storage of biological sample in a really low temperature. Cryopreservation has 2 main methods of slow freezing and vitrification. Both of these methods needs a chemical solution known as cryoprotectant agents that I'm going to call them CPAs throughout this presentation. For a slow freezing, we need really low concentration of the CPAs, and that can help us to prevent internal cellular ice formation. But it's not able to prevent extracellular ice formation, as you can see, which is a problem. Because forming the ice outside of the cells can destroy the matrix or scaffold or structure that keep the cells together. And due to this problem, organ is not functional anymore. On the other hand, the second method, which is a vitrification, can prevent both internal and external ice formation. However, it requires really high concentration of CPAs, which they can be really toxic in higher concentration. For vitrification, we have to make sure that CPA mixture are able to penetrate inside the cell, because during this process, cells gonna lose water. And instead of the water, the CPAs should be replaced. Otherwise, the cell shrinkage gonna kill the cells. And also, we have to make sure that these CPA are not too much toxic, because our goal is keeping the cells alive in the organ. And lastly, we want to make sure that we are able to make the glass state-like, because, as I said, forming the extracellular ice can destroy the organ. But having the glass state can assure us that there is no ice to damage the cells. The good news is, recently in 2023, a research lab was able to cryopreserve the rat kidney for 100 days and transplant it back into the different rat. And they find out this kidney is completely functional. But the question here is how we can extend it to the human body. You may not know, but human organ is bigger than rat and rabbit organs, which comes with 2 challenges. For a larger organ, we're gonna need even higher concentration of CPAs that I said they can be really toxic. And also the cooling rate gonna be much slower, which means this toxic chemicals gonna expose to the organ for a longer time. Both of these challenges emphasize on the importance of having the CPAs, which is less toxic or non-toxic. To address this, we look to the PubChem database, and we find out there is a 9 million chemical out there that they have a similar property to the CPAs that they've been used as a cryoprotectant so far. However, the problem is only about 40 chemicals have been explored as a CPAs. To find out more about these chemicals and potentially finding the better chemicals, we use the high throughput techniques, which they are 100 times faster than the conventional methods. We developed 3 different high throughput techniques The first technique is making sure that we are able to create the glass state-like which prevent ice damage. The second technique is making sure that these chemicals are able to penetrate inside the cells. And the last technique is making sure they are not too toxic during the process. You may be wondering, if I want to test this 9 million chemical, even using this high throughput technique, probably I'm never gonna graduate. But promise, I'm not gonna do that. The plan is gathering lots of information by testing many of these chemicals and then training them to the machine learning algorithm and trying to predict more chemicals from the pool of 9 million chemicals. And I think this method is going to help us to find the CPAs which is less toxic or non-toxic. And finally, I believe my research is going to help to save the life of the people that they are waiting for organ transplant. Thank you.